The Michigan Bean Commission welcomes you to today's episode of the Michigan Bean Academy webinar series. We are delighted that you could join us for delivering benefits from the bioactives and plant-based foods, learnings from the North Carolina State University's Plants for Human Health Institute and the NC Food Innovation Lab. And this is presented by Dr. Ann Lila, Director at the Plants for Human Health Institute, North Carolina State University. And this is Barbara Ivins, Registered Dietitian, your host for the webinar. The Michigan Bean Commission is pleased to offer a series of free accredited webinars, many with a plant-forward eating focus that cover a broad range of contemporary nutrition and food topics. Webinars are a blend of research, science, and practice to help nutrition professionals stay informed on recent developments on relevant topics. Webinars are funded as part of a 2020-2021 USDA grant to the Michigan Bean Commission. A few webinar logistics. A handout of today's presentation is available to download at the Michigan Bean Commission website in the Health Professional section. And the Continuing Education Certificate is available to download after the webinar at the same website as well. Dr. Lila will answer questions at the end of the webinar. Please submit using the Q&A feature on your computer screen. So we welcome today's Michigan Bean Academy faculty, Dr. Ann Lila. Dr. Lila is the director for the Plants for Human Health Institute, David H. Murdoch Distinguished Professor at the Department of Food, Bioprocessing, and Nutrition Sciences at NC State University. Dr. Lila's research focuses on the bioactive compounds found in some foods, particularly fruits and vegetables that confer human health protection when ingested. Dr. Lila's research is aimed at identifying those bioactive compounds and metabolites and understanding how they work. She is particularly interested in compounds that appear to help counteract chronic disease and promote endurance. As a result of Dr. Lila's efforts to locate and study plants around the world, called bioexploration, her research has taken on an international flavor. She has undertaken extensive research projects in Egypt, Central Asia, Oceania, Mexico, Ecuador, Chile, and Sub-Saharan Africa, and is Vice President of the Global Institute for Bioexploration. In 1999, Dr. Lila won a Fulbright Senior Scholarship to conduct research and outreach in New Zealand. In addition, she has served as the U.S. correspondent for the International Association of Plant Biotechnology and as president of the Society for In Vitro Biology. She is also a fellow of the Society and served for 4 years as associate director of the nationally acclaimed Functional Foods for Health program. In 2010, Dr. Lila was named the first David H. Murdoch Distinguished Professor at NC State University. Dr. Lila is an affiliate faculty member at the University of Alaska Fairbanks, serves on the editorial boards for annual reviews in food science and technology and nutrition and healthy aging, and serves on several industry science advisory boards. In 2020, Dr. Lila was awarded the Babcock Hart Award through the Institute of Food Technologists. This award honors an IFT member who has attained distinction by contributions to food technology which result in improved public health through nutrition or more nutritious foods. It is with great pleasure and with delight we are welcoming Dr. Lila to today's webinar. Upon completion of the webinar, participants will be able to describe the broader benefits associated with non-nutritive plant bioactive compounds, be able to explain the connection between plant genetics, agriculture, and food processing, and their role in enhancing content of nutrients and bioactives in plant foods, be able to discuss the type of ingredients and processing technologies being leveraged to make the next generation of plant-based foods, and describe how North Carolina State University serves as a critical partner to drive this innovation. Dr. Lila, I can't thank you enough for joining us today.
Well, thank you much as well. I'll just start to share my screen now. Can you all see it? Okay. Well, if you can see my screen, I'm really appreciating the chance to talk to you all today. And given where we are at this point in time, it's 2021, we're still all reeling from this alternative reality that has taken over for the past 18 plus months, and we're still unsure where it's all going to end. I wanted to put this whole subject of the general public's emerging interest in functional foods or foods for health into perspective. And really the impetus behind the public's interest in functional healthy foods is based on those foods' ability to protect health, and particularly to protect immune health. And why do we have this particular focus? And for some reason, my slide doesn't want to go forward. There we go. The reason is this 400-pound gorilla in the room, and that's COVID. And COVID has taken over, and it is a crisis that has really shaken the paradigm on whether or not consumers trust science. So scientists are coming under a lot of flack now because of all the misinformation or conflicting information about COVID and about how to prevent it and how to eat well and how to eat for immune health. So these things are going back and forth. Consumer priorities for nutrition and advice and products and exercise, this has all changed because of the pandemic. And consumer views on nutrition in the context of a globalized planet. There's been so much controversy about the borders, about what we want to come across our borders, where can we get our food from, is it still safe? So all of this has really entered into the general public's perspective of how we eat and how we eat for health. Now, immune protection is really controlled by both diet and exercise. This is what your grandmother always told you. But the real take-home message of the current research is that you have to have both at the same time in order to really get the benefits of healthy foods. You can't overreach or overdo your exercise, and do 2,000 push-ups. You're going to degrade your immune health if you try and do that. On the other hand, you can't sit there on your couch and eat all your broccoli and expect it to compensate for not moving. The broccoli is not going to even get into your system, and we'll talk about this as we go on. The broccoli is not even going to get into your bloodstream if you're not moving. One thing doesn't occur without the other. Athletes, as you know, can be severely compromised. So, that's not enough by itself. A friend of mine, actually probably my best research partner, David Neiman, he's in Appalachian State University, and he's in the Exercise Performance Lab here in the Plants for Human Health Institute. He wrote a very good in-your-face kind of article last year about COVID-19, a toxin to our aging, unfit, corpulent, and immunodeficient society. And that's a toxin, not like a poison toxin, but a TOCSIN, meaning like a bellwether or a warning bell saying, hey, America, wake up. You're making yourself particularly vulnerable and immunocompromised because of our miserable lifestyle choices. So this article makes an excellent point about how our risk factors for infectious diseases in general, like older males and obesity and related comorbidities, hypertension, type 2 diabetes, and aging, these all lead to these negative changes in immunity, and that really only dietary intake and regular moderate physical activity will together improve immunosurveillance and our ability to withstand viral pathogens in particular. So not high exercise workouts, not bodybuilding, but moderate physical activity. So now let's talk about these plant bioactives and how they do protect our immune health. And what are they? They're not necessarily nutrients, they're not proteins and carbs and fats, and they're not micronutrients like vitamins and minerals, but they are usually smaller molecular weight compounds like flavonoids or alkaloids, carotenoids, glucosinolates, phytosterols, and a few others that in smaller amounts will act directly to your health and travel after you eat them down to your gastrointestinal tract and are transformed by your gut microbiome into active smaller molecular weight compounds that surge back into your bloodstream. So almost always they're going to at first interact with the human gut microbiome, not carotenoids so much, but most of your other phytoactives will first go down to the gut microbiome before they surge into the bloodstream and do you all the good. So these bioactive compounds are exactly what we study, what we are all about here at the Plants for Human Health Institute. And this is our institute right here in front. I actually took this picture when they had a helicopter at a kind of a fair on campus, and they had a helicopter with one of those holes in the window that you could stick your camera lens through. So the Plants for Human Health Institute is all about this. Our mission statement is leading the discovery and delivery of innovative plant-based solutions to advance human health. And we're like 8 different universities here on this campus, all concerned with nutrition, human health, but all coming at it from a different perspective. And Plants for Human Health Institute, we're looking at the plants and the active compounds that protect health, fruits, nuts, beans, vegetables, etc. So we're here on the North Carolina being the eastern coast of the USA. And we're right here below south. So we're near Charlotte, North Carolina, right below some of these powerhouse biotechnology institutions like NCSU main campus, Duke, and Chapel Hill and Wake Forest, and and USC down in South Carolina. But we're here trying to put it together and reach out to the whole world with some unique messages about plants and human health and what the power can be. Now, our focus, as I mentioned, is on these specialty food crops. So fruits, vegetables, tree nuts, herbals, functional foods, beans, all dried or processed forms of these foods, ingredients and supplements as well, even phytopharmaceuticals, but mostly we deal with the food and food products that maximize the health benefits that are our take homes from these fruits and vegetables. And we're very serious at our institute and at our campus about plants and their ability to impact human health. Now, if you look here, this is the core laboratory, the kind of the centerpiece, 311,000 square feet of our campus, the core laboratory. And if you look at this dome on the top of the core laboratory, you're looking at it. If we go inside the core laboratory and we look up into the dome, you'll see it's all painted with a beautiful mural of fruits and vegetables. The eagle is there because the founder of the campus, that was part of his family crest. But fruits and vegetables that impact human health, that's what it's all about here. Now, when I looked at the objectives for this webinar, the first one in the pre-publicity, the first one was, what are the broader benefits associated with these non-nutritive bioactive compounds? And again, they're not nutrients, they're not carbs and fats and protein, they're not vitamins and minerals that are essential to healthy growth, but instead they're secondary compounds, smaller molecular weight usually. There's no recommended daily allowance for these things, but they do have a significant biological effect in the human body. When you ingest these compounds orally, or you put them topically on your skin, because the skin is also a huge portal for health. When you do either of these things, the compounds, after going down to the microbiome and being catabolized and making little metabolites, they'll surge through the bloodstream and they'll interact with human therapeutic targets to inhibit chronic disease or improve metabolic performance. If you think about what we're looking for in food crops, I mean, how did food crops evolve, from 10,000 BC until current times? And initially, once we figured out farming practices, the initial focus was how to increase yield, how to increase the calories that we get out of foods in our field. And then once they were discovered, how to increase the essential nutrients in the basic crops that we grow. And it's really much more recently that health has come into it, phytoactive compounds that were not discovered until relatively recently. And until now, farmers and breeders weren't deliberately selecting crops for health. Let's take one of those compounds, though. One of those compounds for health would be the flavonoids. And the flavonoids, they're a polyphenolic compound. They have a couple of different general classes, flavonoids and phenolic acids in polyphenolics. And you look at flavonoid intake as it's linked to a decrease in all-cause mortality, and it's huge. If you look at the typical US intake of flavonoids, it's about 250 milligrams per day, and less than that if you don't drink tea, because tea is a huge contributor. So it's like 2 fruits or vegetables a day. But look how things drop. Look how your mortality is improved if you just eat twice that. And all you need for 500 milligrams of flavonoids is just a cup of tea, 1 apple, 1 orange, half a cup of blueberries, and that's not nearly enough in my opinion, and a cup and a half of broccoli. So it's almost nothing, and you actually improve your longevity and your health span dramatically just from flavonoid intake, something that your doctor doesn't necessarily tell you, but well documented in the literature. So we know that, and again, we're looking at polyphenols here because that's the one phytoactive that we work with the most, I think, at this institute. Polyphenols from a simple functional food like green tea, like fruits or vegetables, they are known to attack so many different human therapeutic targets that it seems almost ridiculous. It's like, you know, you consider a blueberry the little blue pill that does everything. It's almost like that. I mean, mortality, cognitive function, cardiovascular, we know it improves exercise performance, and I'll tell you a little bit about that as well. It's almost incredible, but why? How is it? Why does the plant compound, the plant phytoactive that's in our food, why does it have that many benefits for health? And it's because they don't act at all like the pharmaceuticals. USA, we tend to be a very pharmaceutically oriented society, unfortunately. And pharmaceuticals act with a single active ingredient. You can do a high throughput screen to test one enzyme getting knocked out by that pharmaceutical. They'll have a very defined dosage and clearance, and plants don't act that way at all. They have multiple phytoactive compounds. Usually they work best if they're working together, not just one magic bullet. They have interactions between those compounds. And a lot of times the metabolic target and the metabolites are still undefined. We're getting better and better with our instrumentation of figuring out what they are, but they're still mostly undefined. Now, if you think about how we discover an active ingredient, let's just say we're looking for an active drug, a really good painkiller, and we might take a plant, you know, in a bark. Like if we chew on bark and we have a toothache, we know that there's something in there that kills pain. So we'll take that compound and we'll separate out different classes of compounds. We'll look for that activity and then further separate it out and further separate it out until we have that big bang, this emphasis, this real bioactivity that's coming from a certain compound, and that's going to be our new drug. And that's how that works. Now, plants don't work that way at all. Usually, if you take a plant extract and you're looking for an anti-diabetic compound, and you bioassay and you see here's the anti-diabetic compound, and you're going to further separate out, you know, purify it some more. Usually you kind of lose it. You don't have anything after you've purified too much. And a lot of times we find out with plants, what we have to do is put things together back again from the plant before we have that activity jump back on us. And that's how plants work. They work with compounds together. So again, phytochemicals are these non-nutritive chemicals in foods, quite a wide variety of them in different foods. Flavonoids probably the most common, polyphenolics the most common, and flavonoids the largest class. And if you look at the 40 top foods for polyphenols, in milligrams for a typical serving, there's quite a variety in different foods that you eat. Now, beans actually are right in there. Black beans, let me see, black beans have 59 milligrams per 100 grams of polyphenolics. And interestingly, white beans have 51 milligrams per 100 grams. So they're actually chock-a-block full of rich polyphenols, and there's evidence for a multiplicity of benefits from these polyphenols. In most cases, the polyphenols are associated with color. White beans are kind of an exception because they're full of polyphenols, even though they're white. And animals use color as a health indicator as well. We'll get to that in a little bit. So color, the different colors that you see in the grocery store is a pretty good biomarker for health. If you just eat your colors, you have a pretty good shot at getting carotenoids, anthocyanins, other polyphenolics, alkaloids. You have a pretty good shot at getting them if you're eating a lot of different colors. So don't have the typical British diet of just brown and white, eggs, potatoes and meat. Try and eat your colorful foods. You'll get your carotenoids from the oranges and reds. You'll get polyphenols from the flavonoids, the blues and reds, glucosinolates and chlorophylls from the greens. You'll get it all if you eat your colors. Now, interestingly, I'm showing a nice, colorful produce aisle in a supermarket, but hey, we have actually changed our food so much in catering to the modern diet, and particularly the American diet, that we have screwed things up quite a bit in terms of the active compounds that plants have to offer. Look at the russet Burbank potato. It's bred. Well, why do you think it's bred? Why do you think they wanted to do a russet Burbank potato? Because it's nice and uniform, grows to a nice shape, nice and pure white inside, and it makes good French fries. So that's pretty much the largest variety of potato that we eat. It's planted everywhere. Now, if you look at the original land races of potatoes, and this is from the Potato Germplasm Repository in Peru, the potatoes were all different shapes. Some of them would be very difficult to peel, as you can see. But inside the potato, it'll be red. Inside the potato, it'll be yellow or purple. That's how potatoes are in the wild races. We've just sort of bred all that away to make it easier to cook with, more uniform. Carrots, now there's nothing wrong with orange. I'm not trying to diss orange here, but all of our carrots now have been bred to be mild flavored and orange and fit into recipes just like that. But the original carrots were a nice purple. Actually, these are coming back in the grocery store now, but for a long time they were just bred out of everything that we wanted. And this whole concept is called One Health. And One Health is a principle that takes into account the interactions of humans, animals, the surrounding environment, plants, and how they affect one another. And the same phytoactive compounds that a plant uses to protect itself, to survive in the wild, those are the same compounds that when you eat them or put them on your skin, that they will interact with your human therapeutic targets to resist disease or to improve your metabolism. So color is an easy biomarker. That's all you really need to think about. And I'm talking about real colors in the wild, not those artificial colors that they put on Cheetos, but the real colors in the wild. And color is actually a beautiful, beautiful biomarker for health. Animals use it. We should use it more too. This is picking blueberries in Alaska. I do a lot of work in Alaska, actually. And the colors in the wild Alaskan berries on the tundra are so intense that when you pick them, your hand's going to get all stained like that. It's going to get all messy because it can't help it. The blueberries are chock-a-block full of the pigments in the skins and all the way through the berry. Do you know that cultivated blueberries, and I'm nothing wrong with cultivated blueberries, I'm not saying that, but cultivated blueberries in the United States, because it's a United States fruit, they've actually been bred not to have the intense color on the inside. If you cut into a typical grocery store blueberry, it's going to be white or yellow on the inside. This was purposely done to breed that out so that they wouldn't stain your hands, and so they wouldn't stain the other fruits in a fruit salad or bleed too much in a muffin. So we actually bred out intentionally the most health protective compounds that berry had to offer. But that's a typical Alaskan wild berry. Now, when you're exercising, there's that expression that, you can't just, take a piece of paper and do, what do you call it, push-ups or lifts with the piece of paper. It's not going to do you any good. The expression, you've got to really take a heavy weight, and do it enough reps that you feel it. So the expression is no pain, no gain, right? You have to feel it before you get the benefits of that exercise. So a similar expression for polyphenolic compounds for plants in the wild is no stain, no gain, because that is when you get the real phytoactive content, the stuff that really helps your health. It's in the pigment a lot of the times, not always, but a lot of the times. And it's a very good kind of a neon light that you don't have to be a rocket scientist to figure out if a plant is got some health protective compounds, because it's right there showing you. If a food has health protective compounds, it's showing you. Now I'm going to show you the kind of the metabolic fate. When you eat, let's just say we're eating a berry or a pomegranate or some grapes down here. When we eat that, it's going to go from our stomach all the way down our gastrointestinal tract, and it lodges in the colon. So it's all the way down at the bottom of the gastrointestinal tract. And in the colon, that's when, I'm showing an anthocyanin here, it gets converted by the bacteria in your gut. So you have 10 times more bacteria in your gut than you have cells in your body. So you have more bacteria in your body than your own cells. And it gets converted by those colonic bacteria to simpler phenolics that'll go up and usually go up and get translocated through the portal vein into the liver, and then eventually go through phase 2 conjugation. Exercise makes a big difference here in terms of the speed of this process. And then finally, what happens is it's going to get released into circulation and exert those bioactive effects. So that's when things surge into your bloodstream and give you the anti-inflammatory properties, the antioxidant, the antipathogenic, the immune regulatory properties. It's only going through your bloodstream after it's gone down to the colon and been catabolized. And exercise tends to accelerate that transport. So what I'm saying is, and most people don't really realize this. They're just starting to, even scientists are just starting to realize this in the last 10 years. It really doesn't matter how wonderful the compounds are in your fruit or vegetable. What matters is, does it get down here and can it be catabolized by your bacteria? Because if not, you're just losing it in your waste stream. And that leads us to the hidden fruit and vegetable quality characteristics, and that'll be biopotency and bioavailability. Now, biopotency, we've been talking about all along here, and that's the strength of a phytochemical, a phytoactive compound in a fruit or vegetable to prevent disease or to improve your metabolism, the power to improve human health. It's going to depend on the content, like how many peaks are in an anthocyanin, what's the diversity of all the different phytoactive compounds, because remember, they don't work alone. It's not a magic bullet. You have to have a lot of diversity. And then also the concentration. How strong is that concentration in a fruit or vegetable? But then also the bioavailability, and the bioavailability is what amount of these phytoactive chemicals actually get absorbed and enter human circulation. It doesn't matter what's in the fruit or vegetable if they're not going to get absorbed and enter human circulation. So is the phytoactive compound bioavailable? Means, is it delivered to human targets? And it's really the proportion of whatever the good stuff is in the produce, how much of it is going to get absorbed and into circulation. And it's going to depend on digestion, absorption, and metabolism. We're not going to get into that here, but that's what it really depends on. And that leads to your second objective of this webinar. What are the connections between plant genetics, agriculture, food processing that enhance those nutrients and phytoactive compounds? And that leads us, and we always have to remember, it's not just it's the content, yes, but also the bioavailability. It's got to be there. So if we eat a blueberry and we know that blueberry has to go all the way down to the colon in order to get catabolized and shoot back into the bloodstream, and we can find a food ingredient, okay, like this happens to be juice or extract from berries, combined with a protein. This happens to be whey protein here. Combining these two into a aggregate colloidal compound. It's like a flour, like a purple flour that actually combines these two and gets it all the way down without degrading, all the way down to the colon where the bacteria can work on it. Well, if you're a scientist, you're going to be very happy about that because you've made a big deal. But if you are a normal consumer, you probably don't, you're not thinking about that at all. No consumer's thinking about how much of my food is getting down to my colon. Certainly not. They're thinking about how much of this stuff do I have to eat to really be healthy? And that really leads us to this project that I'm going to talk to you about. And that is one of the biggest grants that we got in this institute in the beginning from the Foundation of Food and Agriculture Research. It was just four of us, four of our professors here at the institute, and it was closing the gap in delivery of fruit and vegetable benefits. And we had a lot of industry partners here. And the problem is that only 13% of the population really consumes what they're supposed to eat. I mean, you saw those people in the last slide. They didn't look like they were very excited about their vegetables. Most people don't eat what they're supposed to. So how do we close the gap? How do we find strategies that'll get more of what's good in a fruit and vegetable down to the colon where it can be metabolized in the proper way? How do we cook? Do we blanch? Do we juice? How do we prepare something so that it's going to get down to the colon? So we did this by expanding the breeding strategies to develop genetic biomarkers that were going to improve bioavailability, get those fruits and vegetables to the state where it's going to hit our human therapeutic targets, getting technologies like ingredients that were going to make sure that the most good stuff gets down to the colon, gets into your bloodstream, and looking at some clinical trial as well in this. Okay, so why do you not want to go forward? Okay, we had three model crops that we used. We used blueberry because that's probably the best known functional food. Everybody knows that. Banana, the second most consumed fresh fruit in the US, and spinach being a leafy green. And we wanted to look, well, let's just start. We looked at, genetic or breeding strategies. We looked at ingredient technology, and we looked at nutritional equivalency. But let's just start with those genetic phenotyping, the genomics part of it, because that's really a unique part of this, is to combine the phenomics and the genomics. And we've developed a lot of cool techniques in this to make it easier, because farmers are not normally out there looking for genetic markers. They're looking for breeding and what doesn't get insects. And, so we got access to a huge germplasm collection here. We found a way to take a very boring, slow lab technique, which is HPLC, to look at the active compounds, the anthocyanins in berries, which takes a very long time, 77 minutes per sample. And we were able to do it using near-infrared spectrometry. Very cheap technique, can be used even in the field at 1.5 minutes per sample. So those are the things, making it easier for rapid analysis of some of these important genetic traits. Now we looked at blueberry fruits and ploidy, and we were able to differentiate the quality traits in blueberry by their ploidy, by their genetic composition. There's very complicated bioinformatics involved in here. We're able to separate out different blueberries. And interestingly, to make a long story short, a couple of the blueberries that had the highest content of anthocyanins, proanthocyanidins, active compounds, were not as bioavailable, did not really get into the bloodstream, as well as some medium blueberries that just had different textures and other quality traits that made them more bioavailable after being eaten. And we did some really high-tech, high-density linkage map construction to look at quality traits in blueberry and look at exactly how we could correlate the genetics with the amount of phytoactive compound and how much of it would get into the human bloodstream after eating, because that's really the important part. And good solid research leads to other good solid research. So following that one, actually we're still in the last year of that project, we got another one. This one was 13 million award from the USDA Small Crops, Specialty Crops Research Initiative. And this was just looking at the vaccinium, so blueberries and cranberries. Again, genomic resources, DNA markers, molecular genetic resources, and how does that impact fruit quality, leading to bioavailability. And we did this also with spinach, leafy green, linking bioavailability to the health relative compounds. And the highest cultivars in health relevant compounds weren't necessarily the ones that were the most bioavailable. And that was seen again and again and again. And they used a really rapid robotic, and this is Mario Ferruzzi's lab, a rapid robotic digestion model to very quickly look at hundreds of different accessions of spinach and how well they digested and how well they could be absorbed. And a lot of it depended on the leaf texture and the other qualities of the leaf. So many other things like that got involved in the whole thing. So health impacts from these fruits and vegetables are really dependent on the consumption of products that have high nutrient density, but also bioavailability. So if we look at the second tier, the second objective of this project here, can we use ingredient or processing technology to generate some consumer relevant products that are acceptable? Because nobody wants to eat a whole vegetable drawer full of broccoli, right? So can we make products that are acceptable and tasty that'll get the broccoli phytoactives or the spinach phytoactives into the bloodstream, because that's really what we need. So that leads to your third objective in your webinar here, is what ingredient and processing technologies can help craft the next generation of plant-based foods. One of these is combining active compounds from plants and from fruits and vegetables with edible flour. We've used soy, we've used rice, we've used whey, we've used hemp, a bunch of different edible flours, and we combine those phytoactives, and what it does is it actually protects the compound as it goes through the gastrointestinal tract. So making ingredients that helped. And what we used, actually to make it even more eco-friendly or sustainable, is we tended to use waste products from the juicing process. So waste products from grape juice or blueberry juice, in this case, blueberry wine, grape wine. The waste product is pumice, which is all the skins and the fibers and the seeds, and very little of the pulp is left because it's gone to juicing. We found that if we can take this and extract those active compounds, which are the same as what's in the full blueberry itself, and get a very concentrated extract that we then combine with protein to make a colloidal emulsion, or not really an emulsion, but a colloidal aggregate that will be more bioavailable, because that's what it's all about. So you get more bang for the buck, more good out of a smaller volume of food. We don't want to have to eat volumes of things to get a little bit of good stuff. We want to eat a small amount and get everything that we can possibly get out of it. So it's all about making ingredients that capture the best. Interestingly, if we take just blueberry juice or blueberry extract, not combined with protein, we'll look at and this is pre-digestion, and we'll look, it's got all these different anthocyanins. That's what's characteristic. That's what makes a blueberry such a powerful health protective compound, because it's got all these peaks. It's wonderful. But if we digest it, if we take it through the digestive process, it's going through the gastrointestinal tract, you lose so much of it. You see, I mean, in the red line here, you've lost a lot of the peaks. It's degraded severely. Not much is getting to your colon where it needs to go. But if we take that blueberry extract and we combine it with protein, we make those aggregates, and then we put it through the digestion process. So this is post-digestion. Then those beautiful peaks are still there. The concentrations are still there. It's true for blueberry, true for muscadine grape. The bioactivity is going to still be there. Same thing for spinach. We did the same thing where we had the bioactives either delivered as spinach juice or spinach juice combined with protein in a colloidal aggregate particle, which is very, very stable. Gets down to the gastrointestinal tract, all the way down without degrading in the acids of the stomach or enzymes, and it improved the bioaccessible content, especially with soy. And that, so we're really reacting to a food industry that's been undergoing quite a bit of change. Kind of watching my time here, so we're doing good. It is a shifting industry. There's so much more emphasis on health. There's so much more emphasis on private label, local produce, niche growing, consumers wanting local but convenient. A lot more emphasis on health and wellness, and green, so the sustainability, like using pumice rather than using actual blueberries to make a product. And that leads us to your fourth webinar objective, and that was describing how North Carolina State University is a crucial partner to drive innovations. And this is based on the fact the North Carolina Food Innovation Lab, which is labeled up here, is our new pilot plant, research facility, outreach component, but it's all dedicated to plant-based foods. And you can see that the interest in plant-based foods has absolutely skyrocketed recently. Some of this is actually due to COVID too, but it's absolutely gone so high in dollar sales just immediately. And that's what we're all about. So the Plant Science Challenge for us is to use our Plants for Human Health Institute coupled with North Carolina Food Innovation Lab. And then also we have a Plant Sciences Initiative on main campus in Raleigh, all getting together to try and overcome complex challenges of plant-based foods. And NC-PHIL, which is the short name for the North Carolina Food Innovation Lab, is state of the art. It's got all the bells and whistles of a pilot plant facility that has the latest in food processing equipment, and every bit of the equipment, the extractors, the spray dryers, the microwave assisted extractions, everything is focused not just on doing it quickly and cheaply, but on retaining the phytoactive content and bioavailability of everything we put through this lab. So it's a huge emphasis in the industry now, and NC-PHIL is uniquely geared toward, let's just look at how we can get the most out of every fruit and vegetable. It is a new facility. It's not student oriented like a lot of other universities' pilot plants. It's for small entrepreneurs and the big guys in the industry, as well as the research faculty here. And they've got everything. They've got all kinds of processing, thermal, although very low on thermal because thermal can hurt the phytoactive capacity of a lot of things. So they go low on it or find ways around it. Best kind of extraction to get the most bang for your buck, extrusion, drying, particle sizes. They've got everything. It's beautiful. I really hope some of you can come visit, because it is a beautiful facility to look at, and wow, it just kind of blows your mind to see how much is dedicated here, how much is not like the processing plant of even 5 or 10 years ago. Every bit of it is focused on preserving phytoactive content and enhancing phytoactive bioavailability. And so what this has done is it's really taken North Carolina, and we grow a lot of different crops in North Carolina, quite a different variety. But the problem is North Carolina has always grown their crops and then shipped them to another state in order to be processed. Well, I mean, we'll sell fresh produce here, but for processing, it's normally gone out of state. And this is the emphasis of keeping our processing here in state, value added, and to have the most healthy canned, frozen, dried, or other technology products on the market that there could possibly be. That's what we're all about. So you might think, and I'll end with this, you might think that, okay, what have I been telling you for the last 45 minutes? I've been telling you, eat your fruits and vegetables. They're good for you. And that's just what your grandmother always told you. But it's really a whole lot more than that now, because now, for the first time, unlike your grandmother, now we know exactly how it works. We know the transport of a consumed phytoactive compound. We know how it traverses through the body. What's the clearance rate? How can we retain that phytoactivity? How can we maximize? How can we get the most out of an apple or an apple pie? How can we get the most? That's what we're all about here. And I think it's going to be to betterment. It is already to the betterment of our food supply, making things convenient, tasty, sustainable, but also the most health protective that we can possibly do. That's all I had to share. I will stop sharing my slides. I believe that's what I'm supposed to do now. And I'm happy to answer any questions.
Great. Thank you so much, Dr. Lila. That was absolutely fabulous, fabulous. If anybody has any questions, please enter them in the Q&A box. And I will start with one for you. Do you think we will ever see daily values for flavonoids or phytochemicals?
I'm sorry, I was getting a phone call.
It never
Sorry, say it
fails to-
again. Say it again.
Do you think we will ever see daily values going on
Oh.
nutrition labels for flavonoids or phytochemicals, or what do you think that would take?
Actually, your buddy Mario Ferruzzi has been working on that really hard
Okay.
with the U.S. government, with some of the agencies to try and to make those proofs available. The evidence is robust. Now, really, in the last 10 years, we've had so many clinical trials that have emphasized health benefits of phytoactive compounds. So we're finally getting to the point where we'll get enough evidence that will even satisfy the U.S. government and the global governments. Although I think they're much more savvy to this in Europe and Asia than they are in the U.S. But we're getting there, and yeah, things are looking good.
Good. Are there foods or drugs that are consumed that hinder the bioavailability of phytoactive compounds?
Right. Yes, there are. There are definitely are, and some things will compete with each other. I can't. Sometimes fibers, not to say don't eat your fibers, but some fibers can actually compete with the phytoactive compounds. The fibers may bind to the phytoactive compounds. Some proteins, and I mentioned technology to bind phytoactives with protein, but some proteins can actually slow down the absorption of a phytoactive compound. Not that that's always such a bad thing, because if you think about it, if you're a bodybuilder, you're going to want the quickest possible absorption of a phytoactive compound so you can run faster and lift more weights. If you're trying to lose weight, you're going to want a slower absorption, as long as it gets absorbed, but you're going to want it to be slower. So it's not always a bad thing when there's that competition. But yes, there are interactions that will, because we eat a lot of different things. So there are interactions that will compete.
Okay, great. How does the protein colloid phytoelement interaction with the gut microbiome break down to get to the liver portal vein in the bloodstream?
So in the microbiome, in the colon, the protein and the phytoactive compound disassociate. It is a stable colloid, but it's not a covalent bond. It's a non-covalent bond. So you will actually get dissociation with the enzymes in your system, and then so the protein's off to do its good in the human body, and the phytoactive compounds all free and intact at the colon level, which is great, because then that intact phytoactive compound will be eaten up by the gut microbiota, and those phytoactive compounds come out. That's when they go back through the portal vein to the liver for phase 2 conjugation, and then they get into the circulation.
All right, great. How do you see these protein plus phenol compounds used in foods? Will they be be additives for consumers at home, or do you think they'll be incorporated into actual foods?
Yes, we really like it because it's a food, because it's not doing anything crazy to the fruit or vegetable. It's not genetically engineering them. It's not changing them in a molecular way. It's just simply taking, you know what it is? It's taking advantage of the natural attraction between polyphenols and proteins. This is old as the hills. Now, you know what the oldest application is?
Tell us, share.
No guesses, huh? And tanning leather. So you had your animal hide, which is protein. They put the polyphenols from trees, usually tree bark, on it to make the leather nice and flexible, right? That's tanning leather. It takes advantage of the fact that when the animal hide, which would become very rigid and brittle, combines and bonds with those polyphenols from the tree bark, it's a stable binding, so that the texture actually changes and you've got nice supple leather. So we're just taking that same principle of proteins binding with polyphenols and applying it to food. Now, sometimes with food, when proteins bind to polyphenols, it's not a good thing. Like if your beer gets cloudy, you're making beer and it starts settling out, that's a protein polyphenol binding. So that's not a good thing. But if you do it on purpose, because you want to have a stable compound, well, then you've got something that can be used. And you asked, how are they used? So a couple things, and we've just just recently came up with this technology. It's just been a few years. The one that's really out there now, it's called, there's a company called Ripe Revival. And Ripe Revival is taking healthy proteins, like from sweet potato or carrot or beet, and some of these phytoactive compounds, and combining it with blueberry phytoactives or grape phytoactives, and they're making gummies. So they're making high protein gummies. Now, the nice thing about taking a active compound like cranberry juice and mixing it with the protein is it modulates the flavors. So if you have a cranberry soy particle here, the cranberry flavor is modulated, so it doesn't taste all awful like intense cranberry can be if it's really concentrated. It tastes more like pomegranate. It's modulated. And then the soy loses its beanie flavor because of the compound. And this, I'm not just saying this. We've had this stuff put out for sensory analysis with Sensory Spectrum, which is a professional sensory analysis firm that actually has an outlet here on the campus as well as in New Jersey, and they've done repeated studies on this, and they've seen that it does modulate flavor in a pleasant way. So you can have a gummy chock-a-block full of protein, and the flavors are good without sugar, period, just a little bit from the fruit. And mothers feel that that's a sneaky way to get protein into their kids because they think it's candy. They've made smoothies, they've made bars. We worked with NASA because they were looking for a bar that was going to make it for 3 months, and stay supple because of that protein polyphenol binding. It was going to stay supple for a 3-month mission to Mars without bar hardening that's going to break the teeth of the astronauts. We work with the US Army because they were interested in how can we, get fruits and vegetables into the rations in Afghanistan where fruits and vegetables can't be stable. We work with Bill and Melinda Gates to bring some of these things to Zambia, where they have a lot of fruits and vegetables during the rainy season, but during the 6 months of dry season, they've got nothing. They have no refrigeration. They don't have fruits and vegetables. They just subsist on dry cornmeal, which they call nishima, kind of like grits, because they don't have fruits and vegetables. So if we can have a stable source, a shelf-stable source that can just be mixed into their nishima, they've got the fruits and vegetables that they need.
Great. Is protein the only carrier you are looking at that can be combined with phytoactive nutrients to keep the bioactivity?
No, I mean, that's what we've started with, and we've done a little bit now with collagen, because some of the applications are for skin, with algae, spirulina and chlorella. With the algae, which is also kind of a protein, but you can bind with that. It's a very different compound, and fibers. So you can bind with fibers. Also some complex carbohydrates, but we haven't done much with that because there's not a lot of health benefit mostly. But yeah, so you can do a lot of things.
Okay. I assume research as to the benefit of varieties of plants that are studied will influence what growers choose to plant. Do you think that's true?
Well, right now right now it's tough. So these 2 projects that I mentioned to you, the one for 13 million and the one for 2 million, those are both aimed at how can we aid the breeder and the farmer to incorporate more health benefit without having their eyes glaze over because too much science, right? So we want to make it easy. And so that's the point of these is to find easy, user-friendly ways to incorporate this into production. But growers are only going to do it if they see a cost benefit. We see in Europe a huge interest in buying the healthiest berries, the healthiest vegetables. And they look at that, and they will buy, they'll buy a clamshell of blueberries according to the cultivar name, and they'll look for that. Here we don't. You go to your local Kroger or whatever, Wegmans, whatever your grocery store is. You go to your local grocery store and you can buy a clamshell of blueberries, but it's not going to tell you even what kind it is.
Yeah.
Sometimes it'll tell you where they were grown, but they won't tell you what it is.
Yeah, that doesn't help. Then a question on thermal degradation. It's frequently extensive in foods. How does cooking or canning influence retention and bioactivity of these
Well,
phytoactive compounds?
yeah, so that's a real good point because usually usually you associate phytoactive degradation with thermal input. How did I get so popular? So usually when you cook, you lose. Except, like if you cook a blueberry pie, you lose some of the phytoactive compounds in those blueberries, but you actually are going to, it's going to get into your bloodstream quicker because it's become more bioavailable. That's one of the bioavailability criteria. The heating actually improves bioavailability. But in most cases, you're losing, which is why the North Carolina Food Innovation Lab is all about thermal techniques that are short duration, like continuous multi-frequency microwave, that you're bursting microwave to sterilize or to pasteurize a food without cooking it and cooking any of the goodness out of it. Like if you cook some green beans and they become gray and soggy, you've cooked all the good stuff out. But if you can cook them and keep them crisp and green, the good stuff is still there.
Great, thank you. Well, thank you very much, Dr. Lila. That's all the time we have for questions right now. The Michigan Bean Commission was delighted to have you. You've given just a dynamic and very informative webinar. So today's webinar covered, next slide, please, the broader benefits associated with non-nutritive plant bioactive compounds, the connection between plant genetics, agriculture, and food processing, and their role in enhancing the content of nutrients and bioactives in plant foods, ingredient and processing technologies being leveraged to make the next generation of plant-based foods, and how North Carolina State University serves as a critical partner to drive this innovation. And a few reminders about today's webinar. Both the CEU and the handout of today's webinar are available to download at the Michigan Bean Commission website under the Health Professionals section. A recording of today's dynamic webinar will also be available to download in a few days at the same website. And if any questions, please send it to mbc.beanacademy@gmail.com. Thank you for taking time to join us today. And we'd invite you to join us for our next Bean Academy webinar, which is Reasons for the Disconnect Between Dietary Recommendations and Legume Consumption Patterns in U.S. Adults. Presenting that webinar will be Dr. George Bodie out of Oregon State University. And that is on November 4th, 2021. You may now also sign up for that and register for that on the Michigan Bean Commission website. So, and the last slide is please take a minute at the end of this webinar to let us know how we're doing. There'll be a brief question. And again, Dr. Lila, thank you so much for your time and your dynamic perspective. It was truly an enjoyable afternoon. Thank you.
Thank you.